Hi, I'm Steven with Alberta Urban Garden.ca. This is a home garden field trial video. So last time we spoke about the Brix Meter and how it's probably not the right piece of equipment to measure the nutrient density of our food. And in that I promised to try to answer some of our questions using lab results. So in today's episode, what we're going to do is we're going to go through some of the soils results to really see what's going on here. In order to get a really good understanding of what these soils results mean, I've interviewed a number of different professionals in the area, and what follows is our consensus on the analysis of this information. In order to take the samples, we selected the same location in each bed, removed the mulch layer, and took approximately the same volume of soil. After labeling clearly which bed we took the samples from, we put them in a cooler and brought them right to the lab. I would like to take the time to give a special thanks to Maxxam Analytical Laboratories for assisting not only in running the samples, but for analyzing the results. On our video summarizing last year's total harvests, we had a few people ask about the pH of the soils to explain the results this year. If you'd like to see the results, click here now. pH is the scale that defines how acidic or basic the soil is. The University of Vermont reported that the optimal pH for plant growth is between 5.5 and 6.5, when both the beneficial bacteria are operating at peak efficiency and the nutrients are most available to the plant roots. Question 1. Is the pH being impacted by these products, causing the unbalanced harvests? So you're right. pH can have a huge impact on the plants that we grow, on their vitality and their overall production. Too acidic or alkaline, and they're not going to grow correctly. So what we've done is we've actually measured the pH in the soil to see if something was up. pH of the control was slightly more acidic at 5.91, rock dust was 6.0, and biochar 6.17. What does this mean? All three results are within the optimal growth range for pH. And the pH is not statistically different enough in any of them to impact both the harvest and the plant growth. pH has a strong effect on the nutrient availability. All of the values are well within the optimal range, allowing both microorganisms and the nutrients to be more readily available. And this should not have a negative impact at all on the trials. Speaking of nutrient availability, a number of people have asked if rock dust and biochar may impact it in the soil. Nutrients have to be in a form the plant can take up and use. Plants acquire most of their nutrients from the soil, with the exception of carbon dioxide. Generally speaking, nutrients plants need in large volumes are nitrogen, phosphorus, and potassium, or NPK. So question 2, do these amendments impact the nutrient availability in the soil? Nutrient imbalances and deficiencies can have a disastrous impact on both the plant health and production. We have the bioavailable nitrogen, phosphorus, and potassium measured, and we have some very interesting results. First, nitrogen. Nitrogen comes in two different bioavailable forms, nitrate and nitrite. Nitrogen is an essential building block for both amino acids and DNA, which is the cell structure and memory bank. The combined nitrogen availability in the control was 1.4 parts per million, the rock dust 9.7 parts per million, and the biochar on top with 12 parts per million. We can postulate that the amended beds have in fact more nitrogen than in comparison to the control. A good rule of thumb to make sure it's statistically significant is that the difference and the overall quantity is more than 10 times higher than the detection limit of the test itself. The increase of nitrogen in the rock dust amended bed is a little hard to explain, as none of the products claim to actually add nitrogen into the soil. With biochar, it's a little easier to explain, because what biochar does in its product claim is it's able to hold those nutrients in place and prevent leaching. More analysis is definitely required to get a better picture of why these results are showing up. Phosphate plays a huge role in the development of DNA and RNA, which is basically the genetic information of your cells. It also plays a huge role in the development and use of energy through what's called adenosine triphosphate, which is the powerhouse of our cells. Bioavailable phosphorus in the soil had the following results. Control was at 922 parts per million. Rock dust had 1/3 of that at 300 parts per million, while biochar had the most at 1,400 parts per million. The total metals assessment allows us to take a look at both the bioavailable and unavailable forms in the soil. The same trend was observed with the rock dust measuring just 2,600 parts per million, the control 10,000 parts per million, and the biochar 16,000 parts per million. Using the control as a baseline for the concentrations of phosphorus, something in the rock dust bed is allowing the phosphorus to actually leave the soil at quite a bit of an accelerated rate. This could be explained by uptake of the phosphorus into the plants. However, that doesn't explain why the biounavailable phosphorus is also leaving at an accelerated rate. Biochar product claim to be able to hold nutrients in place. When in comparison to the control, this may in fact be true, as the control does not have the biochar in it, allowing slow leaching of the phosphorus over time, whereas the biochar is able to retain more in the soil. Future analysis in the tissue of plants will allow us to know if the plant is actually uptaking the phosphorus, but this still doesn't explain to us why the biounavailable phosphorus is actually leaving the system as well. Potassium, also known as potash, is extremely important in intercellular transport within our cells. In concert with sodium, it's able to actually help us import and export things from our cells. Without it, that import and export just doesn't happen. Potassium is also known to have a key role in protein synthesis, as well as allowing for the opening and closing of the stoma to allow transpiration of your plants. It's also known to have a key role in photosynthesis. The control came in at 1,800 parts per million, the biochar at 1,700 parts per million, while the rock dust had less than half of that at 820 parts per million. The total metals results have rock dust being the lowest at 2,300 parts per million, the control at 2,700 parts per million, and the biochar at 3,200 parts per million, which are not statistically significant differences. What does this mean? Something is taking up the bioavailable form of potassium while leaving the unavailable form. This could be explained by the plants being able to uptake it more efficiently. The tissue analysis will give us a better picture of what's actually going on. So in summary, the pH doesn't seem to be impacting the trials at all. While the nitrogen availability is higher in the amended beds than the control, but the rock dust has lower phosphorus and potassium rates, which may lead to long-term problems. The biochar amended bed seems to be the best balance for pH, nitrogen availability, phosphorus, and potassium. For a full transcript and the posted lab results, go to www.albertaurbangarden.ca. On the next episode, we're going to take a look at the total metals for both the soil and the tissue and compare to see where some of these things may be going. Following that video, we're going to compile this year's 2014 home garden field trial results from all of the participants. Should be very interesting to see what the combined results are. If you have missed any of the home garden field trial episodes and would like to catch up, please click the link on the screen. Thank you for spending time with me today. I appreciate it very much, and I hope you have a fantastic day.